Welcome back, fellow mitochondriacs, to another episode of Cancer as a Mitochondrial Metabolic Disease. At this point, we have now covered all of the major endogenous antioxidant systems. We talked about glutathione and that system, the thioredoxin, as well as the peroxyredoxin systems, as well as catalase. And although there is at least one more system that I do want to cover, which is heme oxygenase, I'm going to do it when we specifically talk about iron metabolism in cancer and the iron-dependent cell death called ferroptosis, because it will dovetail nicely into that. However, today, I want to take the information that we have laid down in this channel, the importance of glucose and glutamine, but instead of looking at it from a bioenergetics perspective, for example, ATP through either cytosolic substrate level phosphorylation or mitochondrial substrate level phosphorylation from a different perspective of redox homeostasis. And then I'm going to explain why Seyfried in his wisdom added oxidative therapies to his classic press pulse graphic. So without further ado, let's get into it. So I'm going to start with a very, I would say, famous paper of Seyfried's that was published in February of 2017. And it is titled Press Pulse, a novel therapeutic strategy for the metabolic management of cancer. And what I highlighted here was the general concept can be applied to the management of cancer by creating chronic metabolic stressors on tumor cell energy metabolism, press disturbances, that are coupled to a series of acute metabolic stressors that restrict glucose and glutamine availability while stimulating cancer-specific oxidative stress pulse disturbances. And before I go to the classic graphic that associated this paper, I'm going to hit on this graphic that they published much more recently in November of 2024. And what we see here is we see glucose metabolism and glutamine metabolism and all the things that it can do. Now, we have picked apart this graphic over and over again. And a lot of times we focus on glucose being fermented into lactic acid, looking at the energy perspective, the ATP that's made here, or glutamine being fermented into succinic acid and the ATP that is generated from that. Or sometimes we've looked at it from fatty acid synthesis or nucleic acid synthesis or whatnot. But we're going to specifically look at how glucose and glutamine contribute to redox homeostasis within cancer cells and how that sets up how this graphic shows the oxidative stress therapeutic here in the form of HBOT or hyperbaric oxygen therapy. So as the paper abstract and the paper suggests, there are press strategies, things that you do all the time, a chronic therapeutic intervention. And then there are things that potentially are pulsed it acutely in a way to disrupt not only energy production, but redox homeostasis. And glucose and glutamine contribute to that. And then HBOT, again, is added here as kind of the final lever to pull, potentially, to upset the delicate balance that is trying to be maintained by these cancer cells. So to start off this particular video, I'm going to talk about this paper here that was published in August of 2021. And it's titled Cysteine Transporter SLC7811 XCT in Cancer, Ferroptosis, Nutrient Dependency, and Cancer Therapy. And it says recent studies revealed that SLC7811 overexpression promotes tumor growth partly through suppressing ferroptosis, a form of regulated cell death induced by excessive lipid peroxidation and iron. So what I'm trying to show here is basically this graphic, or this graphic maybe, in a slightly different way. So we see here that these cancer cells are both glutamine dependent and glucose dependent, and they both contribute to the redox homeostasis in different ways. So for example, glutamine is uptake 10 to 30 times more than normal cells. Glutamine is converted into glutamate by an enzyme called glutaminase. This is where Dawn blocks. This is where several other repurposed drugs and or supplements have at least control over the potential expression of this enzyme. Glutamate is sacrificed to bring in the rate limiting amino acid cysteine into the cell so that all three, both glutamate, cysteine, and what's not shown here, glycine, contributes to glutathione production that helps balance redox homeostasis by decreasing reactive oxygen species and allowing the cancer cell to survive. Glucose is also responsible here. Again, similar story, different graphics. We see that glucose is brought in also 10 to 30 times more than a normal cell. Glucose will divert some of its flux from making energy through substrate low phosphorylation into what is known as the PPP, or the pentose phosphate pathway, which is going to help create a molecule, or regenerate a molecule, maybe a better way to say it, called NADPH. This is a critical molecule for redox homeostasis, which I will show you later. Now, when there is low amounts of glutamine, there is low amounts of glutamine uptake, low amounts of glutamate being created. See how there's multiple dots here, and there's less dots here. And then there's less GSH, or glutathione, that's created. There's less ability to handle redox homeostasis, and you're left with anaplerosis defects and cell growth arrest. So because glutamine can be used to make other things, and what's not talked about here as much, because this is maybe not as in line with Seyfried as some of the other papers are, it's not talking about how glutamine is contributing to cell growth from a energetic perspective, but I will digress here and say that it's going to lead to a growth arrest of these cancer cells if glutamine is restricted. And in the same way, if glucose is restricted, then we don't have as much available glucose to perform, to take part in the PPP and make NADPH, which recycles all of these redox chemicals, which then leads to other types of cell death, but ultimately will also cause major strains on the system. So you can see how both glucose and glutamine are critical for cancer specifically to maintain this balance. Now, I will show you additional evidence to support this later in this video. So I'm going to highlight, again, as this paper was talking about, SLC7A11, another paper that was published in July of 2024 called SLC7A11, the Achilles heel of the tumor ? And I've gone over this paper several times, but I really like this graphic because it really hammers home what we're talking about here. So glutamate is being converted from glutamine, is what allows for glutamate to be present. Glutamate plus glycine plus cysteine equals glutathione. So that's why SLC7A1 is so important, because we have so much glutamine and glutamate available to a cancer cell that has unlimited amounts of those things, or high amounts of those things, that glutamate can be sacrificed just fine and allow for cysteine, which is a more rate-limiting amino acid. It's not as ubiquitous or readily available. So it will sacrifice glutamate to bring in cysteine, and then cysteine, along with glutamate and glycine, which can be made from glucose, if not available in the tumor microenvironment, will contribute to glutathione. And this is a big deal. Now, the other systems that we've talked about, the thioredoxin system, the peroxyredoxin system, also require NADPH as part of their systems as well. Catalase was another system that we talked about, which is responsible for removing hydrogen peroxide. It has been shown to be about 50% of the amount that a normal cell has, but it also requires NADPH. And this is not a topic that has been necessarily explored and forgotten about. The next two papers are very recent, May and June of 2025, respectively. This one is titled Redox Balance in Cancer in the Context of Tumor Prevention and Treatment. And it says malignant neoplasms constitute a substantial health concern for the human population, currently ranking as the second leading cause of death of mortality worldwide. In 2022, approximately 10 million deaths were attributed to cancer. The projections estimate that the number will rise to 35 million in 2050. Consequently, the development of effective cancer treatments and prevention strategies remain a primary focus of medical research. In this context, the impacts on the redox balance are being considered. The objective of this study is to present the current knowledge on oxidation reduction process in cancer. This review discloses the intricate and multifaceted interplay of oxo-reductive systems during carcinogenesis, which engenders discordant findings in the domain of tumor prevention and treatment. This study also examines the controversies surrounding the use of antioxidants, including the impact on other therapeutic interventions, which we're going to do an entire video or maybe a couple of videos on of what not to take during cancer, some of which are antioxidants. The review offers a comprehensive overview of the existing knowledge on the subject, concluding that personalized and precision anti-cancer therapies targeting the redox processes can serve as both effective diagnostic and therapeutic tools. And what it says here, the oxidant-antioxidant equilibrium. Redox balance is referred to the oxidant-antioxidant equilibrium, since it encompasses both oxidation and reduction reactions, which entail the donation and acceptance of electrons respectively. In essence, antioxidants serve as reductants within an organism, whereas ROS and RNS function as oxidants. In the reaction of oxidant with the antioxidant, the former undergoes reduction while the latter undergoes oxidation. Therefore, antioxidant recovery mechanisms using other sources of antioxidant capacities are required. The common unit of antioxidant capacity or universal reductant is nicotinamide adenine dinucleotide phosphate, NADPH. The cytosolic function of this dinucleotide is formed in the oxidative phase of the pentose phosphate pathway via NADPH dependent dehydrogenases, 6-phosphate dehydrogenase and 6-phosphogluconate dehydrogenase during glycolysis parallel process. Moreover, enzymes linked to the Krebs cycle, isocitrate dehydrogenase, malic enzyme, and glutamate dehydrogenase, and enzymes of folate metabolism have been shown to be a significant source of NADPH in both the cytosol and mitochondria. We're going to look at that zoomed in a second. These enzymes of particular importance during periods of increased demand for dinucleotide, anabolic process related to cell division. NADPH functions as an electron and proton hydrogen donor and is an integral part of every redox cycle, which is a series of oxidation reduction reactions initiated by interaction between primary antioxidant and the primary oxidant with regeneration of the antioxidant at the end. And what it's basically showing here is this, first of all, it's a balance. You have to have at least some degree of a balance. It's not always going to be 50-50, but it has to be within some ranges, which we've seen in the past. It's not all good, not all bad. If we didn't have this, we would all be dead because this is critical to life. Antioxidants will donate electrons to radical species that have unpaired electrons that are unstable. So these are the oxidants and these are the antioxidants. And it's a continual donation and acceptance of electrons. Now, this is where the kind of interesting paradox is, because reactive oxygen species or oxidative stress is what likely is at least partially responsible for the setting up of cancer promotion and progression. So I'm not going to idolize ROS or demonize ROS, because ROS can also be used to our advantage to provide oxidative stress that is more damaging to cancer cells than normal cells, which leads to cell death of various types. The redox disturbances in cancer, the imbalance of oxidant-antioxidant balance in cancer involves excessive concentrations of ROS and oxidative stress. One of the original papers in the field of the oxidant-antioxidant balance in cancer examined a cohort of 29 individuals with colorectal cancer adenocarcinoma. Cancer tissue was the study material, colonic mucosal samples extracted during the surgical resection of the tumor. In this way, the tumor microenvironment was studied under concentrations of tumor budding and inflammatory infiltration. The activities of NOx, xanthine oxidase, superoxide dismutase, and catalase, as well as total antioxidant capacity, were found to be significantly elevated in cancer tissues comparison to normal colonic mucosa, histologically examined healthy tissue from the patients. Moreover, the levels of oxidative damage products were elevated in tumorigenic colon tissue, including advanced glycation end products, AGEs, advanced protein oxidation products, AOPPs, MDA, malonaldehyde, and 8-OHGG. In addition, it was observed that the activities of catalase and xanthine oxidase in patients increased in correlation with the intensity inflammatory infiltration. Oxidative stress index and MDA observed to be increased in patients exhibiting a higher degree of tumor budding. However, no significant differences was observed in the level of OSI, oxygen stress index, between cancerous and normal tissues. This is critical. The outcomes of the study confirm that colorectal cancer is associated with the enzymatic and non-enzymatic redox imbalances and increased degree of oxidative damage of proteins, lipids, and DNA. Concurrently, the observed changes that oxidative stress within a tumor was managed by antioxidant systems, SOD and catalase, as evidenced by the lack of variation in OSI compared to normal tissue. Basically, what they're showing is that there is higher amounts of ROS, RNS, oxidative stress within cancer cells, evidenced by the increased utilization of these antioxidant enzymes and by objective damage of DNA and glycation and protein oxidation, etc. But the oxidative stress index was not different from the normal cells to the cancer cells. And the reason I hypothesize this is because the severe upregulation of the majority of these antioxidant systems that are inside the body that are powered by increases in NADPH, which are powered by glucose and glutamine effectively, that there is no evidence of this oxidative stress index, which means that it's still balanced. There's higher amount of oxidative stress. There's higher amounts of oxidative damage. in cancer cells, but it's significantly buffered by increases in endogenous antioxidant systems. This paper was published in July of 2025, targeting oxidative stress mediated regulated cell death as a vulnerability in cancer. And it says here, reactive oxygen species, regulators of cellular behaviors, from signaling to cell death, have complex production and control mechanisms to maintain the dynamic redox balance under physiologic conditions. Redox imbalance is frequently observed in tumor cells, where ROS within tolerable limits promote oncogenic transformation, while excessive ROS induce a range of regulated cell death. As such, targeting ROS-mediated regulated cell death as a vulnerability in cancer. However, the precision regulatory networks governing ROS-mediated cancer cell death and their therapeutic applications remain inadequately characterized. In this review, we first provide a comprehensive overview of the mechanisms underlying ROS production and control within cells, highlighting their dynamic balance. Next, we discuss the paradoxical nature of redox systems in tumor cells, where ROS can promote tumor growth or suppress it, depending on the context. We also systematically explore the role of ROS in tumor signaling pathways and reveal the complex ROS-mediated cross-linking networks in cancer cells. Following this, we focus on the intricate regulation of ROS and regulated cell death and its concurrent applications in cancer therapy. We further summarize the potential ROS-induced regulated cell death-based therapies, particularly those mediated by drug-targeted specific redox balance mechanisms. Finally, we address the measurement of ROS and oxidative stress, oxidative damage, and research, discussing existing challenges and future perspectives targeting ROS-mediated regulated cell death in cancer therapy. So I showed some of these graphics, not necessarily to get into the nitty-gritty of all of it, because we already in some ways have in a lot of ways, but I want to show how these antioxidant systems are not working in a vacuum. We see here that oxidative stress or reactive oxygen species are created a lot of times in the mitochondria themselves. And we see that there's a variety of oxidants or ROSs that are created. Here's superoxide, here's hydrogen peroxide, and they are going to be not only created by various places, but they're going to take place in various reactions and then dealt with by a variety of antioxidant enzymes. Here we have superoxide dismutase. Here we have the peroxyredoxin system and the thioredoxin system. We have glutathione and the glutathione peroxide system. We have peroxyredoxins and thioredoxin system directly connected to the glutathione system. And what you'll see if we actually look again, NADPH is very important here, not only for glutathione and thioredoxins, but several of the other antioxidant systems. And we're going to highlight that in a second. And then what also is interesting about this particular paper is that, it expands my understanding as well as our understanding together of all the different ways that a cell can die. I would say that not too awful long ago, the two major ways that cancer cells would die were by either apoptosis, which is programmed cell death, or necrosis, which is kind of unprogrammed cell death that happens more randomly and less cleanly. And now there are many ways that cancer cells can die outside of kind of those big picture items. So here's apoptosis from the extrinsic pathway. This is when the immune system kind of turns on apoptosis. There's the intrinsic pathway as well. But now there is cuproptosis, which is a copper-dependent cell death. There's ferroptosis, which is an iron-dependent cell death. There's pyroptosis, which is a type of cell death that's kind of triggered by inflammation. There's necroptosis. There's paroptosis, lysosome-dependent cell death. There's necrotic cell death, which is what we talked about in the past. And then there's oxyoptosis and pyronthosis. I don't propose to be an expert in any of these per se, although I would say that I probably have a better understanding of apoptosis and ferroptosis more than any of these. But that being said, is that there's a whole host of new ways that cells can die that we potentially can take advantage of through metabolic therapies, which I think is kind of exciting. Now, at the end of the day, what does this paper actually say? It says oxidative stress mediated regulated cell death in cancer cells. Oxidative stress mediated regulated cell death in cancer therapy. Tumor cells, due to their higher basal ROS levels compared to normal cells, normal tissues, are more vulnerable to oxidative stress and induced cell death. Traditional anti-tumor therapy such as radiotherapy and chemotherapy partly leverage this vulnerability by increasing ROS levels, although this is not their primary mechanism of action. Photodynamic therapy, a more targeted approach, induces ROS through photochemical reactions leading to cancer cell death via pathways like apoptosis, paroptosis, and necroptosis. We're going to talk about photodynamic therapy at length as part of this oxidative therapy series. These therapeutic strategies, however, often have broad effects and may disrupt normal physiologic processes. Achieving selective tumor cell death while minimizing damage to normal tissues remains a key challenge. Here, we explore the potential of existing non-cancer drugs to selectively target redox mechanisms to modulate ROS levels and exert antitumor effects, despite these actions not being their primary pharmacologic purpose. This perspective may reveal whether drugs approved for non-oncologic conditions can influence ROS production, which can influence ROS production, might have antitumor potential. And then in conclusion, it says targeting ROS mediated regulated cell death represents a promising therapeutic strategy in oncology, offering both new opportunities and significant challenges. Specifically, since cancer cells exhibit lower threshold for ROS-induced cytotoxicity compared to normal cells, targeting ROS-mediated regulated cell death offers selective tumor elimination and may synergize with conventional therapies. For example, in drug-resistant cancers, ferroptosis susceptibility is heightened, making ROS modulation a promising strategy to eradicate these cells. Furthermore, combining ROS-inducing agents with existing therapies such as chemotherapy could both circumvent resistance and amplify overall tumor efficacy, highlighting the translational potential of redox-based approaches. Additionally, this compensatory upregulation of antioxidant defense systems in tumors provide additional druggable vulnerabilities. Despite the promise, several challenges remain to be addressed in future research. So we're left with, there's a balance, a delicate balance between antioxidants and oxidants. And this balance is pretty much always the case for a living cell, whether it be a cancer cell or a normal cell. It's just that in a normal cell, there's less antioxidants and less ROS compared to a cancer cell, which has got a much higher level, but still is balanced to some degree. And because there's much higher amounts of ROS, it's much more dependent on antioxidant systems than a normal cell is, because the threshold to death is so much closer. So what we are doing with metabolic therapy, again, this is the fourth way of looking at the same problem as this graphic or this graphic or these graphics, etc. There are two ways to cause an imbalance, right? First of all, we want to cause an imbalance only in cancer cells and not in normal cells. So we want to, number one, potentially inhibit the antioxidant systems that are so heavily depended upon by cancer, number one. So we're going to decrease the ability for the cancer cell to scavenge ROS or quench ROS. And then we have the ability to add oxidative stress to the system and increase ROS production, which creates a redox imbalance, which leads to oxidative stress and death within cancer cells and not normal cells. That is the goal. Now, again, we've talked about metabolic therapies specifically from the ATP or the energetic perspective, but I'm talking about the goal of metabolic therapies specifically for the targeted imbalance of these redox systems to lead to oxidative cell death within cancer cells specifically. Now, I want to tie this in using some of the concepts we've laid down today. So this is a fairly exhaustive, but not completely exhaustive list of the endogenous antioxidant systems within human cells. So we've seen several of these before: glutathione, thioredoxin, peroxyredoxin, glutathione peroxidase, catalase, vitamin C. Some of the things we've not talked about are also on here as well. Now, this is what's fascinating. NADPH is used in all of these systems, and it is important for all of these systems to continue to function. And then what's cool is I created this table with the help of ChatGPT, basically. What happens if NADPH fails or drops in each one of these systems? What functionally happens? And we end up with either peroxide buildup, ferroptosis, protein oxidation, mitochondrial failure, rapid hydrogen peroxide accumulation, ferroptosis, et cetera, et cetera, et cetera. Ultimately, all the ways that these antioxidant systems protect the cancer cell, when NADPH in particular is restricted from these cells, this is what happens. right? These are all the things we want to happen for cancer cells, death effectively, right? From various different mechanisms. Then I asked the question, based off of the literature, what we understand about the production of NADPH, I created this table, okay? So here's the typical contributions from each of the pathways that recycle NADPH. Now, this is in flux in some ways, right? So if glucose, for example, is restricted, glutamine could potentially make up for some degree of the difference. Maybe not all the difference, but some degree of the difference. So it's always in flux. There's only a couple different ways that NADPH is recycled. So here is the first and most important way, the pentose phosphate pathway, and that contributes on average 40 to 60% of the NADPH that's created. What is required for the pentose phosphate pathway to function? Glucose. The malic enzyme pathway, 20 to 35% estimated participation of the total pool of NADPH. What does it require? Glutamine. How about this isocitrate dehydrogenase 1 system? Smaller amount, 5 to 15%. What does it require? Glutamine and glucose carbons. There's another lesser well-known one, the folic acid metabolism. Requires serine, glycine, glutamine, or glucose carbons, 10 to 25%. My point by showing you this is that when you actually add up these systems, estimated amounts of where NADPH comes from, it's mostly systems that require glucose and glutamine. Although this graphic, which is beautiful, I love this graphic, I come back to it continually, glutathione is the only system on this sheet or this slide that is talked about that is made from glucose and glutamine. What we can see here is that 60 to 75% of the endogenous antioxidant systems that make NADPH require glucose, and about 25 to 40% require glutamine. And again, these ratios are likely going to be varied, right? If we restrict glucose, glucose will probably upregulate. If we restrict both, though, which is what metabolic therapy effectively is the simultaneous restriction of glucose and glutamine, we restrict almost all effective NADPH creation, which is responsible for all or nearly all of the antioxidant systems in our body that protect these cancer cells, which makes them even much more vulnerable to the addition of pro-oxidant therapies to create cancer cell death. Does it now make even more sense? Is it more crystal clear, I hope, at this point, that the press pulse strategy at Dr. Seyfried created with his colleagues, I don't want to just give all the credit to him, with his entire group, makes a ton of sense. Ketogenic diet restricted, glucose. Stress management is also glucose. Glutamine as the foundations. And then adding in, I don't think it matters that it's HBOT here or IV vitamin C or others that we'll talk about on this channel. My point is that we've set the stage here by taking already vulnerable cells to redox imbalances, and we've cut off not only their energy, I agree that's important, it is critical important, but it cuts off the major ways that these cancer cells protect themselves from that already baseline higher levels of ROS. And then we hit it with what I personally believe is the hammer, the oxidative stressor. I'm not saying Dawn or some other glutamine drug is not an important piece of this. It is a hammer. It's just, I believe, a smaller hammer in the grand scheme of things to this whole process of what we're trying to achieve here. Now, I wanted to give just one example of how this looks like potentially in practice, but mechanistically. So I only put this because I want to show where this paper comes from. It's called Targeting Ferroptosis Regulators in Lung Cancer. There's actually a really interesting paper that was published around the same time called Targeting Ferroptosis in Colorectal Cancer, May and July, respectively. I would imagine it's the same group, Wang, and here's Zhang and Z. Are they both on these papers? Funny enough, no, they're not. But they have very similar graphics, which made me think that they could have been in the same group. Anyway, my point is, I'm going to show you what this looks like. Now, this is lung cancer model. Do I believe that this is going to extrapolate to every other cancer? Yes. Do we have evidence to support that? Yes. But this is a lung cancer model. So don't think, oh, this is Dr. Peavler talking about lung cancer. Click next, whatever. It's not talking about me. This is the lung cancer model. So let me walk us through here. So what the cancer cell is trying to protect against, ferroptosis, okay? This is iron-dependent, lipid-dependent cell death, okay? The cancer cell wants to survive. So it's going to try to protect against this. What causes ferroptosis? Lipid peroxidation. what does it not want to happen? Lipid peroxidation, right? So it's going to set up systems to help protect against this lipid peroxidation and ultimately iron-dependent, lipid-dependent cell death. So here we have XC, also known as SLC7A11, the Achilles heel of cancer. We see here that glutamate, is being sacrificed to bring in cysteine and then allows for glutathione to be created, and then that can be regenerated with the glutathione peroxidase glutathione system. Now, it's not in a vacuum. There's thioredoxins and peroxyredoxins and NADPH, et cetera, but this is just the graphic we have. We see here that there are a whole host of things that can have inhibitory effects on SLC711. I have videos, I don't want to say dozens, but I have many videos looking specifically at this enzyme system and various things that have an inhibitory effect against this particular system. This is looking at only natural things. We see here resveratrol, we see dihydroartemisinin, which is going to also be artesunate, which is also going to be artemisinin. We see sulforaphane, capsaicin, etc. There are many other things besides what this graphic shows. We've seen that in other videos. My point is, by blocking this transporter, now cysteine cannot be brought into the cell, glutathione cannot be created and ultimately used to protect against lipid peroxidation and ferroptosis, right? So we can see here how these metabolic inhibitors could potentially help us here in our journey. Also, another important piece of this is something called the Fenton reaction, which is a reaction with hydrogen peroxide and iron. It also has other metals. I didn't know this, like manganese and copper and other metals besides iron, but iron is kind of the main one we talk about ferroptosis because it's an iron-dependent cell death. But my point is there's this labile iron pool. There's ways, to also deal with this and to enhance this, which we're going to have a whole part of this series talking about iron metabolism, iron overload, ferritinophagy, ferroptosis, et cetera, hemoxygenase. Here's HO1 hemoxygenase. So my point is, this is also in play and can also be kind of tinkered with in a way that makes this reaction happen more often and more likely, which then leads to what, again, the cancer cell doesn't want to have, which is lipid peroxidation, ferroptosis. So there's ways that the cancer cell will protect iron. It will protect iron in a variety of ways. We'll talk about that, one of which is called ferritin, which is a protein that binds iron, which is why a lot of times people's ferritin levels will be high in the blood for people with cancer because of the inflammation and because of this process to help protect iron from helping with cancer cell growth. But my point is that there's ways that metabolic therapy also can affect this, which can lead to ferroptosis. So again, this is the lung cancer model. This model is exactly the same. If you look at the way they talk about this colorectal cancer model, we have things that can inhibit XCT or SLC711. We have glutathione, we have glutathione peroxidase, we have lipid peroxidase, ferroptosis. Only we're looking at colorectal cancer, right? And we're looking at the same thing. Labile iron pool, here's the Fenton reaction, here's ferritin, ferritinophagy, ferroptosis, etc. Colorectal cancer. That's why I thought maybe this was the same group, but it's not. So my point is, that is why that we have this very stepwise approach. I get that IV vitamin C or HBOT or photodynamic therapy or some other oxidative stress inducing tool is the sexy thing in this particular system, or maybe looking at ivermectin or mebendazole or Don or whatever. But this is a system of systems, sadly, that we are trying to break down in a very systematic way, not to overuse the word system, to achieve a very specific objective. And when we jump to HBOT or IV vitamin C or photodynamic therapy, or we jump to Dawn, or we jump to other glutamine drugs and or natural compounds first, because that's the sexy thing to do by taking a supplement or a drug or repurposed drug or getting into a chamber, without addressing the other systems first, you are doing yourself a disservice. And that's why I want to make this as crystal clear as possible that this system exists for a very specific reason. This paper that came out in 2017, in my opinion, is revolutionary in the way it thinks. It's a systems-based, engineering-based more approach to the management and, God willing, the resolution of cancer by combining therapeutic modalities that leverage all of these systems. And if you miss one piece of that, you may have some success. We've seen and heard people talk about being on a ketogenic diet or a carnivore diet and completely resolving their cancer. Those happen. There's people who will take mebendazole and ivermectin or whatever repurposed drug, and they'll have resolution at subtherapeutic doses. Those things happen. But why look for the exception and not apply the more likely rule to your specific situation? So I hope this makes sense. I hope that you see this as valuable, and I hope that this helps you move forward and potentially, if you have to, apply ketogenic metabolic therapy to your specific situation under the care of someone who understands why and how this potentially could be employed. If you like this video, please like it. If you believe this could help someone you love or care about, please share it. And until next time.